And in fact, it was experimentally shown by an Indian who is right now in ISC. He was a postdoc in Germany with someone called uh, Job Decker is the IC guy. This is another Decker. I forgot. I mean, this is Decker is a common Dutch surname. Okay, this is Keith Decker or something. C Decker, K Decker or C Decker, something. Keith Decker. In Germany, it's a biophysics lab. Okay, so they have shown in vitro that loop excludes from cohescent. So this was the loop exclusion model. Okay. And we also learned about transcription factors.
Excuse me, sir.
Yeah.
Yeah, sorry to interrupt, but in the video, it shows that the loop that is being extruded is just in the form of some naked DNA, but uh, it should ideally be as beads on a string arrangement,
Yeah,
right?
yeah. So yeah, it's a simplified simulation thing.
So
Okay.
how
So
does CTCF get over? How does the cohesion get over the like beads? Are they hurdles, the nucleosomes?
I think not. If they are, they are right. So loops may not be formed there. So one thing that you need to understand is that most of the thing that we are working around, or you are learning in this course, or you are reading through the research papers on IC and the International Genome Organization, is highly skewed towards euchromatin. Okay. Now, first thing, that's one thing, and I'll tell you why is that. So, second thing, loops are actually formed in the early G1 phase. They started formed, right? Okay, when you have abundance of transcription, not so in the M phase you will not have these loops because everything is so compact there that only the neighboring regions are interacting out there. Okay, so G1 phase and S phase where you have more permissive DNA, you have these loops. Now, your question is genuine. How much chromatin or nucleosomes are impacting this? We do not know, number one. And the data or the observation that we have, all these studies are skewed towards euchromatin for two reasons. One is that most of the interesting things are happening in euchromatin, number one. Number two, activity, gene regulation for development and other things. Uh, the heterochromatin is poorly resolved in high C. Can you think of why?
Because it is highly compacted.
So, what?
So one cannot resolve when there are too many proteins in close proximity.
No, why can't? What is the problem? What is the problem in the high C steps or experiment or the observation or plotting? What is the problem? Where is the problem? Which step could be the problem?
Maybe non-specific ligation in the proximity ligation step when there is too much of crowding of those proteins.
Non-specific what?
Like ligation would not happen right at the contact sites, but also like
No, I think most of these days we are doing the ligation in situ. You were saying steric hindrance, right? One possibility, correct? But it turns out that that's not the cause. You're right. So let's say there's a solid stuff out there, a lot of proteins, and then you have open chromatin elsewhere. You have sufficient mobility for ligation out there, but here steric hindrance is not allowing it to have the proper ligation. But it turns out that's not the case. Uh, this is something overlooked by the community, and my group, along with another group, we had this observation and we tried to highlight it. And this our research article was going to group of Job Decker and uh, Lieberman et al., who was the high C developer. Repeatedly, we knew this because the way they were commenting and citing their own paper. We had a hard time to publish this because we were highlighting a problem, a flaw in the technique, and that is the digestion. Heterochromatin is not digest properly. Okay, so you use a restriction enzyme and it fails to digest heterochromatin effectively. So there is a huge skew. The high C data is skewed towards euchromatin, not the heterochromatin, and which is why you have other techniques. For example, there is a technique called, in fact, someone asked in the last class also, the other technique, there's something called GAM, stands for genome architecture mapping. So what they do here is, let's say you have a nucleus. Okay, what you do is you take various sections of this nucleus, cryosections that we call. Okay, randomly you can take various sections. And so on. You take various random sections and you isolate DNA from these sections. Okay. Something which will come together in one section more often is likely to be co-localized, right? DNA which is coming in the same section more often will be co-localized. You can, let me try to give you an example. So this is A loci. C loci. And B loci. Okay, inside the nucleus. So you know A and C are interacting. Let's say this is the background information. So what will happen? Let's say you have one scenario, one nucleus section. Okay, so let's say in the one nucleus section is coming like this. This is one section, right? So the co-localized one would come in multiple sections, right? Because they are together. However, the ones which are not co-localized will come only in this one, right? Only in this one, right? So this blue one is coming only in one section. However, the other ones are coming, other two which are co-localized will come in more sections, right? Isn't it? So you collect this data of different sections, okay, and then you go to computer typically. Okay, go to your computer and get the 3D information, get this matrix, spicy-like matrix. Okay. So this method, when people map the chromatin interactions through this method, this is a bit more sparse data. Okay, it's not because of the limitation of how many sections you can make. Okay, so these are all cryosections. Sections. The data is little sparse, but when you compare this data with the high C data, what people observe is number one, there is no, of course, we compared it, high C versus GAN, there is no uh, bias towards euchromatin. Okay, the number of reads, if you look at around this locus, let's say, this is your matrix, right? So you take the sum or the average of each column and plot it here. So in high C, if this was euchromatin, let's say there was a euchromatin here, and this is H chromatin. Okay. What people found is that the reads here look like that. Okay, so there is a skew here in the euchromatin, they are being mapped. The moment you look at, so this was your high C thing. So the moment you plot for GAN, it turns out that there is no such bias. It is like that. Whether it's heterochromatin or euchromatin, coverage is same. So what I plotted here is the coverage. Okay. And additionally, what people observe is that GAN is also resolving the 3D structure of heterochromatin. Okay, which means that there is a pattern now. So if you look at high C data, heterochromatin is just one smear. Okay, let's say, let me try to plot it here, some example. Let's say there is a, this is one tag, this is a heterochromatin tag, this is your euchromatin tag, this one. So you find some pattern, right? This is a loop pattern or blah blah blah. Here you will just find all the dots are just having higher values, kind of smear. There's no pattern as such. The moment you look at the GAN data, you find the patterns there also, heterochromatin tags. Okay, there are differences in high C, and there are more methods. Okay, there are microscopy based methods also. There is another method which is devoid of dilation. I think it's a Sprite method. So, in interest of time, we will not go through all these methods. But this one is clearly telling that we have a bias of heterochromatin in high C. Now, high C has also improved. People are not using restriction enzyme, they are using MNase enzyme, okay, which is giving resolution at the nucleosome level, which means around 200 base pair resolution. So that's one thing. Okay, so the loop extrusion is done, and then about loops thing, I wanted to tell another thing in this class, and that is that some people like Peter Cook, who originally proposed and found at the early observation of transcription factories, he has also proposed that transcription factories could be a unified model for various kind of regulatory elements. And I try to convince you in few of the classes that the distinction between regulatory elements is blurring up as we are learning more and more. And hence a promoter, you see, the difference of these two regulatory elements is blurred up now because enhancers are having promoter-like properties and promoters are also having enhancer-like properties. So there is a unified model based on transcription factory for enhancers, silencers, and insulators. So I'll try to give you the example here. Consider the following. Let's say there are two transcription factories or two, let's say, phase separated globules, okay, A and B, and we have a locus on DNA. Okay, so let's say there is a region X. X is a gene, let's say, and this is an enhancer of that gene. Okay. Now there was this Y gene over here, which was supposed to be localized on B factory, but it couldn't be localized on B factory. Why? Because X was expressed, so it was dragged to A factory by transcription factor or whatever mean. Okay. So one thing that you observe is that E is also localized in the same factory, so there is an interaction here of enhancer to promoter. So if they localize in same factory, there will be interaction. So this is explaining the enhancer promoter interaction. Of course, there will be loop here. I didn't draw the loop. However, it is also telling that somehow these two regions are actually working as a silencer for Y, isn't it? The moment they are activated, they are open chromatin, okay, the Y gets repressed. If they are not activated, they will not localize to A factory, and then Y will go to its factory and gets transcribed, right? So, which means that these are silencers, could work as silencer for Y. Okay. Otherwise, this is an answer for X. Okay. Now, think of another scenario now. Let's say uh, at this particular factory, you have X and Y interacting with each other. There's a loop formation. A factory here, and then you have a B factory over here. Something happens. There is an in-between locus which has high affinity for B factor and it gets activated. So what happens is that this locus has come here now. Let us call it as Z. Z has come here on the B factory now. So it has dragged now Y and X away from A factory where they were making the loop, which means that Now Z is functioning as an insulator for Y and Z, right? And so this is pretty much feasible. This is pretty much possible inside the nucleus. We do not have a direct evidence, but there are reports which says that the same element, same regulatory element like enhancer element can actually function as enhancer in one cell type, and exactly same element could function as silencer in another cell type. So maybe this explains that. Okay. So that's where the transcription factory model is very interesting. It resolves number of the conflicting observations that we see in the literature. Okay, so what else? In the one of the classes that I was teaching, we stopped and I mentioned that show you some data on cell cycle dynamics of these interactions, right? So I have got a figure here from a paper. This is Amos Tanai from Israel with Peter Fraser in Oxford, in England. So they have done some single cell IC and all that. So what you see here is, first of all, you see cell cycle thing, right? Cell cycle thing over here, right? And what you see here on the y-axis is the genomic distance of contacts. Okay, so more bluer means higher contact frequency, light blue means lower contact frequency. What you observe is that in the M phase, which is here and here, you have uh, more short range contacts, okay, or mediocre range contacts, which and the long range contacts which is coming over here are absent there. You see this thing. That's why, that's because you will have interactions of the proximal region. That's why, that's how you can make a typical M phase condensed chromosome, right? This cruciform structure that you see of chromosomes in the textbook is because you don't have long range interaction, but the nearby region are highly interacting with each other. So this is what the nearby regions are, right? They are interacting here, right? We are interacting here. So you don't have anything here, nothing here, nothing here. The moment you move into G1 phase, you start getting these long range interactions, and these are appearing. You see these, which makes TAD-like structure. You see these TADs here, right? So these TAD-like structures are coming up in G1 and S phase. And as you move to late S phase and G2 phase, you short range interactions again comes up and it starts condensing. So this is like not condensed, but not having long range interaction. Condensation just started. So you will have a thin diagonal in the beginning, a thin diagonal. The moment condensation builds up, okay, initially it is like, initially condensation is like that, okay, and then it condenses further over, right? That's how you can see that in textbook, right? More condensation means more nearby regions are started interacting. That's how they are leading to this state, right? And that's what happens here. What happens here? Your diagonal will become thicker and thicker. So that's what the cell cycle dynamics is of long range interactions. We will also see when are they formed in development, right? After fertilization, are they formed immediately? Is there any difference from maternal and paternal chromosome? We will see tomorrow in the tomorrow's class when we talk about development. Now, I would like to stop with this spicy thing. There is sky is the limit. You know, if I keep on teaching you, I can offer you one course on this, but that's not the point. So let's move on with the something else with respect to development only. Uh, I'll ask you something. Let's say that you have chromosome, let's say, and you have certain deletion in this chromosome. Let's say 1 MB. This is Length of this region is 1 mm. Okay. Now, this is the observation by Marcus Pambray long ago in 80s and 90s. Uh, this was not fitting well with the Mendelian genetics. The observation was very strange. They found that there is a chromosome 15 in human, and there is a 1 MB deletion which associates with one disease phenotype, one syndrome. Later on, they saw familial cases where same deletion, exactly same deletion, but the phenotype is contrasting. Did you get this? Very same deletion, two contrasting, I'm not saying two different phenotypes, two contrasting phenotypes. What's happening?
Sir, are they in two different cell types?
Well, it's a genetic deletion. Let's say there is a germline deletion. I said familial cases, which means it has to do something with the germline. Deletion happens in cell type specific manner. Those are somatic mutations. They are not well studied also. If we talk about the common somatic mutations, cancer associated ones are well studied. So if I'm talking about familial cases, it means it has to be, it is there in all the cells. It's a germline mutation. What's happening? It's mind-boggling. When people observe this, there are people coming up with familiar history of the same syndrome, that you have same deletion. Okay, one person is showing one kind of phenotype, another person the same deletion is showing completely different phenotype, contrasting phenotype. What's going on?
Sir, maybe imprinting has something to do with this, depending on the deletion came from maternal or paternal.
Yeah, exactly. So you have two chromosomes, right? So if this is deleted paternally, you have one syndrome. If this is deleted internally, you have a different syndrome, right? So I can tell you what is it. So if you have maternal deletion, not maternal expression, maternal deletion, it leads to Angelman syndrome, given by the author who first observed these symptoms, Angelman syndrome. And if you have paternal deletion, you get Prader-Willi syndrome. So this is popularly written as PWS and AS. What's happening? Two chromosomes are exactly same. You consider, most people consider that maternal and paternal chromosomes are same, right? Then deletion shouldn't matter whether it's maternal or paternal. So what's happening? Somebody said imprinting. What is it?
Maybe depending on imprinting, they have a different architecture, that
What
particular segment.
what architecture?
Like the TAD formations may differ.
Well, deletion is deletion, right? Sequence-wise, they are same. Well, largely 99.99% would be same, right? Sequence is same, right? But this tells me, I think I had given that paper to one of the students. I can tell you that there were, in 50s or 60s, there were, sorry, 70s, there were some classic experiments done by Azim Surani and some other authors also, Bara Forlongi. Okay, what they have done, the fundamental question was, uh, is there any maternal or paternal contribution to the independent contribution to the embryo? So what Azim Surani did was they made a mouse offspring with two maternal chromosomes. Okay, what would have happened?
The phenotype would be abnormal because no paternal
What
contribution.
abnormal? It was embryonic lethal. Not only it died, the placenta size was very, very small. It didn't develop. If it was possible, think about as a layman also. If it's possible to have a offspring from two mothers or two maternal chromosomes or genome, maternal genomes, then parthenogenesis is also possible, right? So there is a barrier to this parthenogenesis, and you know that having parthenogenesis is also not beneficial. Why? Because you lose the opportunity to bring the diversity through recombination, the advantages of recombination in a population, right? So what's happening in the case of PWS and Angelman syndrome? Let's discuss that. So here is the example. This is the kid. And if you search about Angelman syndrome on Google, you'll find smiling faces, laughing faces. Angelman syndrome. This is PWS. And I can tell you what are the phenotypes. These are known as happy puppets. Now there are two reasons. First is that they are always smiling and happy. Their motions are jerky. Okay. So like a break dance kind of motions. So they are happy puppets. They are more often happier. So these guys are relatively short in length and fat. And the common thing is the mental retardation, but different kind of mental retardation. The first thing is that milk suckling thing. It is difficult to feed this one. Poor suckling. You can write one of the phenotype is poor suckling. This is over suckling kind of thing. Contrasting one. This is lethargic or lazy. This one is hyperactive. Hyperactive to an extent and difficult to control this fellow. Uh, Short height and average height. Motor retardation is common in both. They are more of autistic, and they are more of bipolar disorder or psychotic bipolar disorder. What do you call psychosis? Psychotic. Okay, which means little bit of schizophrenic. Yeah. So just to give you some understanding of autistic and schizophrenia, they are contrasting phenotype. You need to understand autistic means they are not interacting with people, right? They consider people as objects, autistic. And schizophrenic people are other way around. They consider even the objects as people, other way around. So to cut it short, the Angelman syndrome kids, they are resource consuming. Okay, so they are oversuckling uh, and very very active and autistic. Autistic people are, you can consider them into a selfish category. Okay, because they are not interested in altruism and other things which comes through the social interaction, while schizophrenic people are kind of seeing interactions everywhere, even with the objects, and they have delusions about peoples and so on, right? So these traits are actually are the traits which are coming from paternal chromosomes, these ones, okay? And these are coming from lack of paternal chromosome and the expression of the maternal genes. Now, why the paternal chromosome is giving an active one and the selfish kind of phenotypes? That's because of genomic imprinting, the way it has evolved. There's a parental conflict theory, and we will get into detail of that theory. It tells you that the paternal genome has evolved uh, in the evolution in a way that it tries to enhance the growth of the fetus or the offspring, right? Why? Because there is a strong competition in the male population, right? There is a sexual conflict kind of thing. Sorry, there is a parental conflict going on now. The males are competing with each other, which we call as sperm competition, right? The male which is actually enhancing the growth of the future offspring would be selected. Okay. While maternal genome would be the one which will be growth limiting. It will tell baby, okay, you grow, but leave some resources for me. Think of in simple scenario, just think of let's say that this is the worm. Okay, if the baby is really big inside, the fetus is really big, and it is keep on growing. This baby is keep on growing here inside the worm. The first thing is that it is taking a lot of resources of mother will become weaker and so on. And such a big baby is difficult to deliver also. How would it come out? Right? You need to have a growth limitation also. You need to nullify the male effect, right? You need to get the appropriate size and save the resources for herself, for the mother. So maternal genome would express the genes. It has evolved in a way that it expresses the gene which are growth limiting. Okay, so maternal growth limiting, and paternal genome has evolved to enhance growth promoter. So this is growth promoter, and all these examples are of paternal nature. Okay. Over suckling, which is telling to increase the growth, the kid is over suckling, is hyperactive, because that's what the male genome tends to. Growth is one broad thing that I mentioned. It also makes the offspring more active, that will be selected in the sperm competition, right, to become more selfish, and likewise. If you lack this, you get contrasting phenotypes over here, okay, which are not the part of maternal genome. Sorry, which is not the part of paternal genome. Poor suckling, it's leaving the things for the mother and so on. Now, some of these things are actually controversial. Not many people agree that these are contrasting phenotypes, but this is one hypothesis. Okay. Now, on this chromosome, in this region, if you zoom into this region, okay, you will find there are genes. Okay, there are genes which are maternally expressed. Okay, so I will. Okay, so maternally expressed, and then we have a set of genes which are paternally expressed. Okay, now there are two regulatory elements in between over here. Control regions. We call them as control regions. Okay, so there is a one element, let's say this white one, this is regulatory, non-coding regulatory, which upregulates the maternal genes. So they are being expressed. And it also silences this region. Exact mechanism is not known. And this region, when this is not silenced, and if this is methylated, the maternal one, you will have expression of paternal genes through this locus, green locus. But these two regulatory regions are reciprocal. They inhibit each other through methylation, promoting the methylation of other. Precise mechanism hasn't been worked out. Okay, so if you have maternal deletion, you will be losing these genes expression, and these genes are not expressed on paternal chromosome, other set of genes and so on. So maternal and paternal deletion will matter, because there's a different set of genes which are maternally expressed and paternally expressed. Okay, they are completely silent. It's not like this is a stochastic phenomena. This is imprinting, which means that it is parentally fixed. Only the paternal allele which is coming from mother will be expressed for a gene which is paternally imprinted and maternally expressed. Imprinting means repression, okay, and vice versa. So if you delete this region, if you have deletion of this region, let's say you just have deletion of this region is gone, basically deletion, then and this deletion is maternal, then the function of only these red genes are gone. Paternal genes will take over now, right? And if this deletion is paternal, then function of these paternal genes are gone, and So the maternal genes will have its influence on the offspring. So this is imprinting, and we will see that with another example. If you have 5 more minutes, I can elaborate that, which is more, which is better worked out example. Can I go ahead? Sleeping. Hello.
Yes, sir.
Okay, so a better example is IGF-2 gene, insulin growth factor. Okay, this is a growth factor well known, and insulin growth factor receptor gene. So this one is growth promoting, and this one is growth limiting. So this would be expressed from paternal chromosome, right? Growth promoting, this will be expressed from maternal. Exclusively in human being, in most mammals. Okay, this is the thing. It is well studied in mice. Okay, now I'll give you the. I'll ask you something. Let's say I make a matrix now. So I say paternal expression of IGF present, absent. And likewise, maternal expression of IGF2R, which actually binds to IGF2. Okay, so it blocks it. Okay, so what will be the mice phenotype? Let's say you have paternal expression IGF2 is present. Okay, and maternal expression, so you will have a normal mice, right? Normal mice. Normal mice. What will happen if IGF2 is absent from paternal chromosome? Okay. However, so present, absent. IGF2R is present on maternal chromosome. What will happen? What will be the phenotype?
Retarded growth.
Yeah, so we will have small phenotype. Tail is a bit longer, but anyway, so okay. Now think of this scenario. IGF2 is expressed paternally, but IGF2R is not expressed maternally. So you have growth promotion, but not growth, no growth limitation. So this means that you will get a big phenotype, right? Overgrowth. What about the last one? Maternal expression of IGF2R is absent, and paternal expression of IGF2 is also absent.
Lethality, probably.
No, it's actually a normal phenotype because there is no growth limitation, and IGF2 is not the only growth factor. You have IGF-1 also and insulin also, insulin growth factor and so on. So people observe that this is actually a normal phenotype. Of course, when I say normal, nothing is normal, right? Okay, which means that you will have some other compromise which appears later on in the lifetime, and if it doesn't appear, then it is affecting the population fitness. So I can tell you why did I say so. uh, when we talk about essentiality of the gene, we say that, well, you delete this gene and this, which is embryonic lethal, is essential gene, and then we have genes which are showing phenotype like this one, small mice, big mice, right? Right. But then there are many, many mutations or knockouts in mice which has no observable phenotype. Many times you people delete a gene and nothing happens. Then they say, oh, there is a backup copy of this gene, a duplicated copy which is taking over the functional. That's not, at least to me, it's not convincing because there's no straightforward evidence of this. In fact, the evidence is the other way around. So what happens is that when you have such deletion which is not giving you observable phenotype, you have to wait long enough to see, okay, the phenotype. Maybe it's affecting the fitness of the of the population in the later time, right? Or that mutation will show effect in certain environment, okay, certain conditions. So every gene is essential in that way. Not essential. Every gene is associated with certain phenotype. It's just that you need to scale up the things. You need to look at the population fitness. You need to look at generation after generation and see what happens to the population fitness if you have this mutation, and you will observe something there. It has been shown in C. elegans, something which doesn't show phenotype in one lifetime. You look after certain generation, you see population fitness is compromised, or you expose the species, your organism, to different condition, environmental condition, and then you see the phenotype. Anyway, so this example of IGF2 and IGF2R is supporting the parental conflict theory. What is that theory, parental conflict theory? I just elaborated giving you the example of Angelman syndrome and PWS syndrome. Is that paternal genome has evolved in a way that it increases the fetus growth. Maternal genome has evolved in a way that it tries to save the resources for herself. Okay. In the next class, we will look at more theories, and this is called genomic imprinting. When you have imprinted expression, we call it as parent of origin effect. Okay, it depends on whether which allele, expression of allele will depend on from which parent it is coming from. So the expression of alleles is parentally fixed. Okay, this is called genomic imprinting, and you have around 300 to 400 genes in human genome which are imprinted. Okay. So there are more theories also, but this is the most accepted theory, parental conflict theory, and I will give you a few more theories which are popular in the next class. And then we will see that how it has what happens to imprinting during evolution, during development. So the next class will be all about development and the role of epigenetic modifications during development. Well, one class is not sufficient to teach you development, but this is what we can offer you for the time being. One of the reasons most of the papers that we are going to present are associated with development some way or the other is this only, that since we couldn't teach you development, which is an important part of the PGNT course, we have given you the papers. And I also feel that we taught this course in a bit lousy manner this time. One of the reasons was we suddenly offered this based on the students' demand. So multiple instructors came together and we compiled this course. But maybe from next year onwards, only one instructor will teach, so that there is a coherence, what is being taught and what should be the flow. Uh, According to me, the development part should be the majority for the epigenetics course. If you have learned about transgenerational inheritance in the beginning from Rajesh, without understanding how epigenetic modifications are regulated or they are regulating the developmental process, you won't be able to appreciate how this transgenerational inheritance is possible at all. In fact, it's not possible. If you look at the developmental trajectories, the marks are erased and reestablished. Then what do we mean by having the memory which is going to offsprings? Okay, so things are interlinked. Most of the regulations that you have learned in various mechanisms make sense in the view of development. Epigenetics is all about environment, okay, and environment plays a major role during development. You have intrauterine environment, you have morphogens and other things which initiates a cascade of epigenetic modification in the whole environment. Okay? And you shouldn't get surprised to look at your teeth and your eye. Okay, they have same genome. Okay, look at your skin and your heart or your liver or your lung. They are very very different, but the genome is same. Is the way they have developed is the thing which is making them so different. Alright, so we will continue with the genomic imprinting and then we will look at the trajectories during development, what happens to genetic modification during development. okay, and where does imprinting lies during that trajectory. If I get time, we will also look at X chromosome inactivation.
Excuse me, sir.
Yeah,
Sir, I have three short questions. Can I pose them now, or?
yeah.
So here in Engelmann, that syndrome, just to confirm, even if we find a way to ectopically express that 1 MB segment, it wouldn't matter, right? It wouldn't rescue the phenotype.
Sorry. Okay, so you're saying that if we express the maternally expressed genes, which are silent paternally.
Yes, sir. The segment that is being lost, if we introduce it ectopically, it still won't improve, right?
Well, no, it will improve. But doing these experiments in human is not possible. People would have shown this in mouse. I'm sure there must be papers around, not for this particular locus, but at least for IDF locus, it would be there. But you need to understand that the genome has evolved in that way that it needs certain copies to be expressed from maternal and paternal chromosome. Okay, so there is a gross inheritance difference how the uh, alleles are expressed.
Sir, that means you're saying if we introduce the missing copy, then how does it acquire its for example, if we introduce the missing maternal copy, how does it acquire a maternal status? Because it doesn't have any epigenetic modifications, maternally inherited
Yeah,
epigenetic
so you
modifications.
need to make sure that those things are expressed at the desired level. By the way, these genes are also dosage sensitive. So even if you are having a external way of expressing this inside, you need to really moderate the level of expression. Otherwise, overexpression is also lethal for dosage sensitive genes. See, you might say that then why the maternal copies are needed, right? Or paternal one. So this leads to, I can give you example uh, where the coordination is needed between the mother and the offspring. You need to understand how it has evolved. Uh, if you have same alleles expressed in mother and same alleles expressed in fetus, okay, there is a better coordination, right? Because alleles are same. You need reciprocal interactions between mother and the offspring. And to give you the example, the suckling thing, if the kids are crying for milk, okay, the mother's mammary gland starts making milk. Okay, if there is no demand of from the offspring to get the milk, mothers don't secrete milk. It is well known in human beings also. So there is a coordination going on between mother and the offspring. Okay, reciprocal one. There could be multiple genes involved, but this coordination would be better if the similar alleles are expressed. And I can give you the example which supports this. For example, all the genes which are expressed in placenta, which is serving the offspring, right, feeding the offspring in utero, all the genes which are expressed in placenta, imprinted genes, are actually maternally expressed. None of them are paternally expressed. So there is something called co-adaptation between offspring and mother. Number one. Okay. Another theory is the cytonuclear conflict theory. Right. So you have mitochondria, right? And you exclusively get the mitochondria from mother, not from father. So if you have genes in the to resolve this conflict, that the genes which are needed for the coordination, energy coordination with the mitochondria has to be maternal. Okay. So here there is no offspring thing. This is in within cell conflict. Take that in mind. You also have a theory which is related to dosage sensitivity that I just mentioned. If you are expressing a gene from two alleles, you can't control their variance. Okay, controlling one allele is actually less cost. It is more cost effective than controlling two alleles. It is dosage sensitive. Controlling the expression level at both places in a way that there should be coordination. You should know that this allele has expressed this much, so now you need to express only this much. There has to be some crosstalk, right, which is not possible. Now you silence one thing, and the balancing thing is only happening at one allele, right? So it's possible that imprinting has actually evolved to minimize the variance of expression. You don't want variance, you want a certain dosage only. And if you have other allele, there is a more variation, overall variation in the concentration. And since the genes are dosage sensitive, it is selected against it. It is only expressed from one allele. We also have hypothesis related to parthenogenesis. It is a barrier, imprinting is a barrier to parthenogenesis. If you have two maternal chromosomes, you don't have a viable offspring, so parthenogenesis is avoided through imprinting. And we know that parthenogenesis is not good when it comes to the combination and the diversity and so on. There's something also related to parthenogenesis, is something called ovarian time bomb hypothesis. It says that there is a trophoblastic disease where, just like parthenogenetic animal, the oocytes without any fertilization start making some tumor-like tissue, start expanding. Okay, to avoid this to happen, okay, this kind of development, uncontrolled development happening. This is called trophoblastic disease, some kind of disease which would have been observed in some cases. To avoid this disease to happen in females, imprinting has evolved. Right?
Sir, and you showed the TAD dynamics during the cell cycle from G1 to M phase. Like, does this mean the architecture is relatively static in terminally differentiated cells, those in G0?
Static means what?
Like, they don't differ, the loop extrusions and all, they don't differ in those cells that do not divide?
Cells that do not divide, I didn't get the question. Why
Sir,
would it be different? The mechanisms is, first of all, it's a hypothesis, right? There's some evidence, supporting evidence, but no direct evidence. My problem with the loop extrusion is following: how much energy do you need? At every loop formation, you need so much of ATP. Do we have that much ATP? Is there any calculation? If you find some calculation, please educate me. Number one. Number two is that if that hypothesis is true, it will be true in all cells. Right? So let's say you are saying that terminally differentiated cells, it is already established here. The loops are already established and they are functioning. That's all.
Sir, I was saying, since they change during cell cycle, the change that we observed, does that exist in different already differentiated cells?
No, I didn't get. Can you say it again?
You can go to that slide where you said at M1 you have a lot of thickness at short range contacts. The distance, the shorter range, you have them, and when you go through the cell cycle, you form long range contacts. The basically diagonal kind of expands. So this whole change, can we expect these changes to be observed in differentiated cells also?
If the cells are dividing, these changes will occur. Yes. This has nothing to do with the development. This is growth.
Okay.
You understand growth and development. Development is differentiation, growth is your proliferation. Right? This is just the proliferation. Any cells which are proliferating will show this dynamics. And this is anyway expected, right? Even in the textbook you have seen that condensing and open thing is the same thing. Personally, I'm not surprised to see what is shown in this slide. It's just that somebody asked in one of the class, and I'm trying to show this. In fact, people have worked out that this thickening happens, and they have shown that how chromosome, the mitotic chromosome condenses. So things are worked out using Hi-C. The mechanism that first this happens, second step is this, third step is this, and so on.
Okay, sir. Got it.
Okay, so we'll continue talking on printing in the next class, and then we will look at the developmental trajectories of methylation and histone modifications. If there is no more question, I'm stopping this class.
Recording.